Hi, this is Dr. J. Bailey, and we're going to be working on topic 1.1 on written answers. This is where you're going to find most of the common questions related to 1.1. So we're going to go over some of the mark schemes, and we're going to see some questions that are so much related to each other. So here we have two questions. The first one, as you can see, they highlighted here, it says outline and discuss. Usually the outline, this is more like about 3 to 4 points. Right? While discuss, whenever you see a discuss, this definitely has to do with at least 7 to 9 points. It depends on the questions and IBs. But why did I put these two questions together? Because believe it or not, if you see the first section of the question right here, it says outline the cell theory, discuss the cell theory. So in the first part of this question right here, the answers will be practically the same thing as outlining with a little bit more of a discussion. So we're going to see some of the things that is common between these questions, and with having them in common in their mark scheme, that means you can have a robust mark scheme. And if you have that mark scheme in your hand, then you can always use the same kind of information to write on your essay. Afterwards, we're going to look at the other section of it, which is just its limitations. But so now the first thing we're going to do is actually focus on the cell theory, just on the first part. Now you're also going to see how different the mark scheme looks like when it says outline compared to discuss. So let's go ahead. Here, let's look at the first sentence of the mark scheme. Okay, here it says for the outline, cells can only arise from pre-existing cells. That makes sense, right? When it comes to discuss the cell theory, here's the first sentence for the mark scheme of this question. It says cell theory is accepted explanation of life. You can see that none of the two have exactly the same answer. That's okay. But you will see as we go along with the extra additional information, you will see that these guys will match. So one of the most important thing is start mentioning the cell theory, which is 3 sentences. Now, what about this one? Well, this was never said anywhere else. Like, this is a logic one. But IB sometimes they come up with these mark schemes out of the blue, like, oh, it's the accepted explanation of life. Yeah, because it is. It's logic that we're talking about how cells are and how the living things is. So sometimes a sentence in IB as a mark scheme will show up and you guys could never imagine. That's why there are some common things that will always show up in the mark scheme, and these ones are the ones you have to focus. For instance, this one right here, cells can only arise from pre-existing cells. That's a for sure sentence will be in any mark scheme related to cell theory. Let's move on. So we go to the second thing that it's in the mark scheme. Here says living organisms are composed of cells, or another way, the smallest unit of life. And then here, organisms are composed of one or more cells. Look how close they are. They're basically saying that organisms are composed of cell. So in these both type of question, even though it's an outline and the one is discussed, they're both asking you to mention this very important thing, that organisms are made of cells. So this part here, composed of cells and composed of one or more cells, should be in any mark scheme related to cell theory. Let's get more going on with this. Now we have the C. Organisms consist of only one cell carry out the functions of life in that cells. Cells perform functional life functions at some point in their existence. Now this is for the outline of cell theory. On this one here it says the cells are basic, fundamental, smallest unit of life. If you look at C for this discussion, it has the same idea as this one, which says that the cell is the smallest unit of life. While on the outline here is talking about how, and let me erase this to be more clear again, it's organism consists of only one cell carrying out all the functions of life. Basically, they're saying that one cell that does everything, metabolism, homeostasis, nutrition, excretion. Now, that means every cell should be doing all of that. Metabolism, homeostasis, reproduction, excretion. That's what it's trying to tell you. Well, this one goes back to talking about it's composed of cell units of life. If you really focus here on these questions, they're asking about cell theory. So far, you can see the main idea is they come from pre-existing cells, right? Which is also founded here, right? And then you have the second one, which is the smallest living organism composed of small units of life. This one here, look, this one. Okay, let me erase here. Let me just show you how interesting the mark scheme sometimes can be. On this first question, as you have it this as just one answer. It says the living organisms are composed of cells and is in the small units of life, but that's just one point. Both of these do information at one point. While in this one, it's interesting because you have organism composed of one cell will be considered as 1 point, and then if you mention the smallest function of life, while cells are basic, smallest function, it's 2 points. Well, here it's just base as one. See, that's your luck. That's your trying to go for IB. That's why IB tries to make it hard sometimes, and that's why you have to just write as much information as you know. You might be lucky that you end up with a mark scheme that gives you this as 2 points, and sometimes you might be in a bad situation where even if you wrote those two and you think those are 2 points, but actually in the mark scheme by being 1 point. That's why I always say when it comes to essays, do not be brief. Write as much as you want because you don't get points for writing so much. You only get points off if you contradict yourself. If you say the sky is blue and then you say the sky is gray, then you are taking points off. But if you talk about everything there is in the sky, like there's birds, there's clouds, there's stars, and you keep talking, you will never get points off for that. Okay? So remember, you only get points off for contradictions. So now that we see this, so far the main ideas, let's go back here. Actually, let's go to the slide right here. So what are the main things that you must write when it comes to talking about cell theory? The first one is do say the cells come from pre-existing cells. Do mention that all the living organisms are composed of cells. Okay, and do mention that the smallest unit of life is the cell. So look at here, the new mark scheme, cells are coming from pre-existing cells. That's exactly like the one that we just did right here, this one. So there's some repetitive things, right? Now, will this first one here, a cell theory is accept exponential life, appears in every mark scheme? No, not really. Your luck. The same thing here, this one right here. Will all the organisms consist of one cell that carries all the functions of life? Not really. But I tell you one thing, I would put this in here without cell theory because it's true. Think, you have trillions of cells. It means every single one of your cells should be able to do metabolism, should be able to do homeostasis, should be able to respond, should be able to reproduce, should be able to excrete. That is what it's trying to say here, that one cell can carry out all the functions of life. And then here it says, although most organisms conform to cell theory, there are exceptions. So this already went straight to tell about the exceptions. Now, it's an outline. Because it's an outline, you just generalize the information. Just say that there are exceptions to cell theory. That is, if it is an outline. But if it's discussed, you'll see that it has its limitations. And here, at this point, I was just focusing on talking about the cell theory. So for the cell theory, most important things that you have to discuss is simple. Cells come from pre-existing cells. All living things have cells. The smallest function unit is a cell. You can say that one cell carry all the life functions. And even if you want, you can also mention here that there are some exceptions of the cell theory if it is an outline. Now, this question usually is a 3 to 4 points. So by you mention the 3 statements of the cell theory, you already got the points. But never stick to 3. If they ask you for 3, put 4 or 5. If they ask you for 5, put a 7. Always put extra 1 or 2 points there because that makes sure that you get the whole points. And then you can see here an addition for 1 right here. It says spontaneous generation in life has been disproven. Where the hell did this right here? None. Do you see this here? It wasn't. So in this paper, God knows which year was this paper, in the outline, there was nothing talking about the spontaneous generation. However, on this paper, I don't know which year we're, so let's say this is maybe was 2010 and this was 2016, whatever it is. So sometimes the mark scheme will come with extra answers. That's why if you're luck, the more information you can write, you can do it. And if you study the 1.1, it did talk about Louis Pasteur and did talk about how he disproved the spontaneous generation because it's all about cells coming from previous cells. So when you mention right here this one, cells can only rise pre-existing cells. If I was you, after you mention this one, quickly mention about how Pasteur experiment basically disproved the spontaneous generation. And this might give you 1 point or might give you 2 points. Then you mention organisms are composed of cells, and then you can say that the smallest unit of light is a cell, and then you can say that just one cell carries all the functions of life. So what you're doing here is you're targeting all the possible things that can come out related to cell theory. However, I haven't talked about the exceptions. So when it comes to outline, you just mentioned there's exceptions, but when it comes to discuss and it tells your limitations, you have to talk a little bit more in details. So we just focus on the part of cell theory, discussing it. Next thing we're going to do now is focus on the exceptions. That means the limitations. That's why if you see this question right here, it is still the same question of the previous slides, but now we are actually focusing on the limitations of it. And this question, the part of limitation, it follows very much with this one, which says discuss the possible exceptions to cell theory. So I'm going to show you 2 mark schemes when it comes to discussing the possible exceptions. And when I say exceptions, it's the same thing as limitations. That means they go against the cell theory process. In this case, let's start. So here is what we get on each side. One side here, it says that skeletal muscles, let's put in yellow here, the skeletal muscle fibers, they are larger, they have many nuclei, are not typical cells. It doesn't mean you have to write those 3 sentences. You can say the skeletal muscle fibers are larger than the usual cells, or you can say skeletal muscle fibers have many nuclei that is not normal like the other cells. All the other cells have 1 nuclei. The skeletal muscle in this case, it has many more. That's one of the exceptions. Look on this side. It says striated muscle cells contain many nuclei, while most of the other eukaryotic cells have 1. Or you can say red blood cells have no nucleus, while the most eukaryotic cells have 1 nucleus. So you have 2 options. Is the mark scheme going to be the same? No. So in this one, they add a little bit more. They talk about how red blood cells is a cell that has no nucleus, while the striated muscle cells have many nucleus. On this one, it just mentions that they have many nuclei. So what would you do? As a combination is, since both mark schemes says about the skeletal muscle fiber, or by the way, skeletal muscle fiber is the same thing as a striated muscle fiber. So either way, if you write skeletal muscle fiber or striated muscle fiber, IB usually takes that as in consideration. But you have to mention that they have a lot of nucleuses. That means they have many of them in just that one cell. And that's one of the exceptions. Another one that they have here is the fungal hyphae. So you can see that this is another exception they have, and they're not individual cells. What does that look like in the drawing? So basically a hyphae, think of a root like this, and usually we will think that it's a bunch of cells like this, right? That these cells are making the root. So the root will look like that, right? That's what it means, but the truth is hyphae does not work. Hyphae is literally one long root, and all the cytoplasms are connected, all the cells are connected. So that means they just have a little gap, but their cytoplasms is actually connected to each other. And cytoplasm means the space inside the cell. So if I had to highlight the cytoplasm, it's all this area here, blue area inside, they're connected to each other. And these little black corners here are just a little bit of the wall, but you still can see the cytoplasm connected. So this is a fungal hyphae. That's why it says it's not divided into individual cells. That means they're all a bunch of cells connected as one. On this one right here, when it talks to limitations, they talk about the giant algae. Okay, they have a complex single cell structure, or in other words, an organism as large as a giant algae would be expected to be multicellular, but they only are made of one cell with one nucleus. Be careful. The giant algae is not like the skeletal. The skeletal muscle is a long cell with many nucleus. The giant algae is just like it. Let me erase this one to give it more space. It's just like it, but the difference is it's a long one with just one nucleus. Now, usually we always say that as the animal getting bigger, it's more likely that we have to go through division, but the giant algae does not do that. So we already talked about two limitations. Actually, if you look here, there's basically three. We talked about the mussel, right? We talk about the fungal hyphae, and we talk about the giant algae. These are three, and actually these are the three ones that you have to know by heart for the IB, right? Again, make sure starting mussels have a bunch of nucleus. The hyphae here is not individual cells, it's a bunch of cells that have their cytoplasm connected. And the giant algae, which is very long, it doesn't divide into many cells, it's just this long, huge cell with that one nucleus. So that's just the beginning, but guess what? There's more that you can talk about limitations. Look what they have here as a mark scheme. Unicellular organisms can be considered acellular. So what does it mean acellular? That means they don't have a nucleus. Oh, then it goes back to that thing where it says, what? That it has no nucleus. You can use that one. And then look at here. You have the asepti fungi hyphae. That's what we just discussed, and you can find in this mark scheme too. But it says tube-like structure that contains no cell membranes between many nuclei. So that means they doing the drawing again, so you understand. Again, imagine this. And basically you have their nucleus here, and they barely have this, like a cell membrane that is basically separating them. They don't have that. They actually have, oops, that was too big. Let me go back. They actually have more likely an open area in which all of their nuclei are basing. It's compared to each other. So basically, again, we have a slime modes, as an example, many nuclei. So the hyphae here is a lot like the mussel, okay? They're just connected. They might have a little bit of that wall here, but the cytoplasms are connected, just like in mussels. Another one. Let's what they say. Let's see what they have here. They say because they are larger than typical carry out functions of life. So basically, they're just saying they're carrying the functions of life, but they're very large. Who does that remind? Right here. Algae. You're just saying it's so huge that usually when you get really huge, you want to split into many cells. But no, this one keeps getting huge and it doesn't split and it still can function. That means it can do nutrition, homeostasis, reproduction, whatever you want. But look on this other one. On this other one here, it has an additional of viruses that you don't see in this mark scheme, but you see it here. Now, viruses might have some characteristics of living things, but guess what? They are not cells. Viruses are not living. So viruses have some of the traits of living. So you think, oh, if the virus has some of the living traits, then it should be living. But that's not true. It's still not a cell. It's still not living. So virus is an exception. It has some of the traits, but still are considered not part of the cell structures. So that's something that you can find in this mark scheme right here, but you don't find it on this one. So again, you're never going to find exactly the same mark scheme. But if you see the general trend, that's what you take. So the general trends so far, what are the general trends that we can see here? To keep in mind whenever you talk about limitations. Talk about the skeletal muscle. Talk about the hyphae, right? Just because you have the muscle here and you have the hyphae. Talk about the giant algae, because that's just talking about typical large cell that does not divide. Then after you talk about those three, why not add virus in there? That's a fourth talking limitation. But guess what? There's more. Here, as you can see, what is it saying? Some tissues or organs contain large amount of extracellular material. Where have you learned this in class? Probably not. But guess what? IB puts extra stuff there and expect you to know. Not every teacher teaches this. Like, how would I know that some tissues or organs contain large amount of extracellular material? It doesn't say anywhere. So usually barely any teacher will be teaching that. But IB will mention. That's your luck. Here on the other side it says, if all cells come from pre-existing cells, where did the first one come from? That's a limitation right there. That's go back to the story of Pasteur. Pasteur was saying, oh, I proved that living cells come from previous cells. But he did not prove what was the first cell. And just mentioning here this idea will be perfect for you to end the limitation topic. Okay? Now, here is the important thing. Because it says discuss, and this is a lot of things what purpose students do, They just list, say, oh, skeleton striated muscles, fungal hyphae, and algae, and stop. No, please write about them. Why are they an exception? Because when you say the word discuss, they want you to explain. And this is where students are so brief. Do not be brief. You got to be a good writer. So why skeletal muscles? Because the skeletal muscles, it has many nuclei within that one cell. Why are the hyphae? Because again, this cell is not divided into many cells. It's the one cell which they has all their cytoplasms connected. Or you talk about the giant algae, which is just one cell that is so huge, but it does not divide like the other organisms. It stays as one huge cell with one cytoplasm, with one nucleus. Then you can mention the virus. The virus basically has some of the living traits, but it just does not mean he's a living. And last but not least, you can finish and say, here's the most interesting one, is that we can use Louis Pasteur experiment and say that cells came from pre-existing cells, but nobody is able to explain where does the first cell come. And bam, you nailed it. So in this way, we already have discussed the cell theory, which remember is those 3 statements. And then the limitation, you talk about the exceptions. So in general, what we have, cell theory would have about 3 or 4 comments that you mentioned, which we did in the previous question. And then limitations, you probably have about 3 to 4. If this all depends on the number of this question. So always, if the question come out to be 9, my friend, be in generals right here. I would write all of this striated muscle, the giant algae, the hyphae, the virus, and this. Look, you get 5 points plus whatever you're talking about the cell theory. So you're combining everything you know, including the Pasteur experiment. So you mentioned the 3 cell theories and talk about the Pasteur experiment. And then you talk about the limitations. You can hit 9, but usually there's more like 8 points. So look at the number next to the question. The more points, the more you're right. Now look at this. Again, we're just putting it all together. We are just seeing that there is more. Look at this one here. It says that vitreous humor of eye, mineral deposit of bones, xylems of trees, and other examples. My friend, this is on the mark scheme, and I guarantee you most of the teachers don't even talk about it, so don't stress about it. So sometimes there will be more information on the mark scheme. You're going to be like, but wait, the teacher was never in the book, the teacher never mentioned, and that's what IB does, is just add some extra information. But there's always, if you notice, there's always a reoccurrent answer in this. And you can see it, I can even highlight it. They're always talking about the striated muscles. They are always talking about the hyphae, right, the fungal. They're always talking about the giant algae, which is the same thing, saying that they're larger than the typical cell. These 3 are always mentioned. And then you can do the additional stuff, which you can mention about the virus. You can mention that we don't know what's the first cell. So if you can, always look for many questions related to the topic and make sure that you see the most common things in the mark scheme, and you will be able to answer it. And guess what? We have more. Yeah, look at this. This one here, let's put the yellow. The statement cell theory, all living things or most tissues are composed entirely of true cells. Who would have thought about that? Right? Discuss the possible exceptions to cell theory. Many students, when they read this, they assume, oh, let me just go straight and talk about the exceptions. Nuh-uh, be nicer. Well, show it to the teacher that you know more. If you want to know about the exceptions, before you talk about the exceptions, my friend, go ahead and talk about the 3 statements of the cell theory. You can get points by mentioning it. So first say, well, the cell theory statement, the cell theory states 3 statements. 1, cells come from pre-existing cells. Cells is the smallest function of the view of the life, and all living things are made of cells. Bam, you mentioned the 3. Then you start talking about the limitations, which I mentioned. The muscle cells, the algae, the hyphae, the viruses. And where did the first cell come from? Now you have a full essay written for cell theory, and there's no way that you will get less points if this question shows up in your test. Now, since we have been talking so much about Pasteur experiment, let's talk about it. This is a question that comes out, and it can come out a lot as a multiple choice or a short answer question. Describe how Pasteur experiment provided convincing evidence to falsify the concept of spontaneous generation. So here in this slide, I first will go quickly to review what is this topic about, and then in the next slides, you're going to see what does this mark scheme look like. What are the things that they look like? So just to be clear, what is the first thing that Pasteur is trying to do? He's basically saying, this is what he proved, that cells comes from previous cells. More important, they have to be living cells. What does the spontaneous generation say? They say that cells, they come from nothing. Like out of the blue, magic, poof, they just show up. That's the difference between spontaneous generation and Pasteur. So what Pasteur did, he based through his experiment, he rejected the spontaneous race. He says, no, cells don't just come out of nowhere. They have to come from a previous cell. But Pasteur was not able to explain how the first cell comes. But you can start by just mention that. What Pasteur experiment proved, disproved, and then talk about this. Next, it says describe. That means when they say describe, they want you to tell the entire experiment. How did they do the entire experiment? When it comes to how they did the entire experiment, it is important that you actually write in details. Now, if you're not a good communicator in writing, sometimes drawing it can help you. So I would draw, for instance, the entire process. I will have the flask here, right? I will have the chicken broth being put here. And then I would say number 1, I will use blue as the microbes are here. On the second scenario, I would have the flask here, and I will have the chicken broth. But this time I will close the flask, and I will definitely put like sterilize here or boil the broth to kill all the cells. So now it's dead. And then I will have the third flask here, in which I will have it cracked soon. I'm just going to put here. And you have the chicken broth, and you're going to say that it has been boiled. But then what did I do? I kind of cracked part of it. And that led me to get back the microbes back in and grow. So you have to explain the process. So in the first process is he noticed that there was chicken broth in the flask and microbes were growing, or bacteria, you can also say bacteria, were growing on the broth. And people thought that those bacteria just showed up at nothing, right? Then the second part he did was heat it up. He boiled that broth. And when he boiled that broth, he basically, before he closed, he tried without closing. He boiled it, and then he left it for a day after everything was dead. And again, things showed up. So on the third example, what he did is he did boil it. And when he boiled, that means now inside here, there's only dead cell. And he closed it. So when he closed it, days later, you didn't see any bacteria inside, nothing grow, which proved it. This one, number 2, it proved that, see, there's dead cells, and see, there's no living things coming. And they're like, that's not sure, right? So when here, this one, he actually proved it even more, because he says, you know what, I am going to crack it open. And he's going to prove that, see, they were like, well, if there's nothing here, how is this guy showing up? He says, well, because he must come from the atmosphere, and it must be something that is living in the atmosphere that comes in. So he broke it, and he proved the living microbes just come in, and they start to grow again, even after all the boiling and breaking up this flask. So this was his experiment. Now, let's see how he puts in words. How would IB want to say? The first thing I would always tell you is explain what spontaneous generation is. So you can see that the first thing says spontaneous generation is life appearing out of nothing, okay? And it has to be from non-living cells. And then here, this second part says cells can only come from pre-existing cell lives. So the first part here, right, is the spontaneous generation, and the second part here is the Pasteur. But guess what? In this mark scheme, mention those two will only get you 1 point. It happens. Sometimes they put it as a B. We don't know. Then the second thing is description. we have the description. So broth or cotumirium in flasks. So basically, this is the drawing that I did. So just say they use broth. They use broth and on a flask. So by just saying they use broth in a flask, you're already getting points for this mark scheme. Look at that. Third, it says here broth is boiled or sterilized. Sterilize means cleaning it up to kill what? The microbes. So you have to mention that the broth was boiled or sterilized. So first you say you put broth in a flask, then you boil or sterilize the flask to kill the microbes. So don't just say, oh, we boiled the flask. Don't do that. You gotta say that they boiled the flask in order to kill the microbes. Then the third, no clouding. What does that mean? There's no sign of bacteria growth, or there was no reproduction, did not appear in flasks that were boiled flask. So that means when you killed, okay, you boiled and killed everything, you didn't see any microbes coming. Then you can't say especially when the flask was closed, right? Because again, once you boil things, you're killing all the microbes. There was no microbes there afterwards because it's been killed. Next, allow bacteria organisms instead of microbes. So yes, I say microbes, sometimes I say bacteria. IB is okay with you mentioning any of the two. But look at this. After the necks of flask were snapped, boiled broth became clouded again. So remember, you boiled, you killed all the microbes, it's sterilized, there is nothing there. And no microbes grew up again, unless it was open flask. But if it was closed, nothing grew up. But if you open that closed one, guess what? It goes back again, even after it's sterilized, it goes back again, as it says, to create new microbes. So we have to describe these steps as I was mentioned here. Now, because microbes from the air contaminated, and you have to mention, you can't just say, oh, I opened the flask and now there's microbes. They're going to say, why? But why is there microbes again? You have to mention these microbes are coming from the atmosphere and landing on the top of the soup, and then they're growing. So you're basically telling a story. You just have to tell the entire story of the experiment. And this is how you will get these points. So it's extremely important that when you are writing anything that is written as describe here, it's like you actually telling story by story about it. Okay? It's very important that you do this. And last, look at this. It says curved necks allowed indirect exposure to the air, but prevented entry of microbes. What does curved neck means? Curves are this. If you have a flask that is like this, it's not curved, but sometimes they have flasks that looks like this. What does that mean? These curved ones, microbes here, they can come in, but they landed here. So it means you are right here at this point exposed to the air, but it does not allow it to go all the way in. So it doesn't basically it's preventing the microbes from fully going all the way to the inside of the flask. That's what's meant by the curved neck. This is not a curved one. So you can see the difference between a curved neck and a non-curve. So you can add this one right there. So a big quickly scoop on how Pasteur experiments provide. First, foremost, mention what spontaneous generation was, and mention what Pasteur experiment was proving cells come from cells. That's the first. Second, mention they used broth in flask. Mention that the broth was boiled or sterilized to kill the microbes. Mention that there was no bacteria growth after they was boiled. Then mention that after the neck flask were snapped, the beans broken, there was growth of microbes again. And where did this microbe comes? They come from the outside. The air was contaminated. And if you need, you talk about the curved necks. They don't allow to go all in. Now, if you can see here, this has 1, 2, 3, 4, 5, 6, 7. It's never 7 points. Usually this will be like a 5 point, but they give 7 points on the mark scheme. So you might hit 1 or 2 things more. But like I said, if it's a 5 points, put a 7. Describe the whole thing. Words are very important too. So that's if you have to explain Pasteur experiment. So the next thing we're going to talk about is the functions of life, which is nutrition, homeostasis, response, reproduction, excretion, right? These are the things that every cell in the world has to do. These are just some of them. So this question says state. When it says the word state, it means it's just one word, my friend, one word. And he wants to know the function of life of Paramecium, and that is carried out by cilia. So cilia, here's a Paramecium, and cilia is found on the outside of the Paramecium. And the job of the cilia is for motion. That's what it is. It just moves it. So we're going to say, how did you know the Paramecium cilia is for movement? First, we know the cilia is on the outside for movement and basically to attach to the walls and stuff like this. But also, IB expects you to know all the functions of Paramecium. So if you didn't, go back and do the studies and make sure you know all the functions of it. It's even on my videos. The second one, it says state the function of life Paramecium, but this case is talking about the contractile vacuole. So that means right here, you have a large contract vacuole, right? And inside of it, you have storage of water and stuff like this. What's the purpose of that water? It's just to keep his balanced out. So it's for homeostasis. What is it exactly doing? It's just making sure that there is a balance of water inside and out. So there's not excessive water so it doesn't burst. So if you didn't know this, go back to Paramecium. Go back to the video, learn about it. So let's look at what the mark scheme says. You can see movement or locomotion. The word locomotion means to move. Another thing is for feeding or nutrition. Yes, the cilia has these little hairs that attach to food and bring it in. But just the movement is the most common one. And here you talk about the homeostasis, because that's what's the point of contract vacuole. It has water there just to balance the water of the Paramecium. So that was pretty easy. Let's go for the next question. So here is outline the method of nutrition. Again, it's Paramecium again, as well as this one outlined one aspect of how the P. caudatum carries out homeostasis. So one is talking about nutrition and the other one is talking about homeostasis. So how would it work? Let's look. Outline, basically it's bullet points and usually can be between 3 to 4 points, right? 3 to 4 points. Now, just going back here, usually these, when it's state is 1 point. It's always going to be like a 1 point thing. So going back here, this is about 3-4 points. This is usually 3 to 4 points. Can it be 2? Yes, it can, but it's barely. It's usually more like a 3-point thing. So let's look at what it works right. So when it comes to nutrition, heterotrophs or consumers, what do? They feed on bacteria, algae, yeast cells, right? So here is the most important thing. They're feeding on it, on bacteria. You can say that. But then my question is, Miss, how do I know about this? Well, you have to know it by heart about it. There's no other way. On this side, what about homeostasis? It lives in fresh water, so the water enters the cell by osmosis. What does that mean? Well, if the cell here, if the Paramecium is here, and it's surrounded by a lot of water, well, you got to make sure that the water, if there's more water coming in through osmosis, right? In the fresh water, why? Because fresh water means that they have less solute. So there's less solute on the outside and there's more solute inside. What's going to happen? Water wants to move to a place of high solute. So when there's too much water is coming in, voila, the saver is the contractile vacuole. What it does, it just spells out the water back out. This way, keep the Paramecium from bursting. So this can be followed up with the next one, and you can see it, contractile vacuole. And you see the word is underlined. When it says underlined, that means I'd be saying this word has to be written. Otherwise, no points. So it collects and expels water, which makes sense. So if you're living on a fresh water, right, water enters as what? By osmosis. And the contract vacuole, what it does, pull the water out so it doesn't burst. Here, consumer or heterotroph, as you wish, what it does, again, it's feeding on bacteria, or you can say it does not have chloroplast. What does that mean? Because chloroplast what it does is photosynthesis. His way of getting energy is by the light of the sun and producing sugar. But since he's not an autotroph, he's a heterotroph, he has to eat other things, in this case, bacteria. So In the end, heterotoph must be qualified, but this is just basically means you must mention that it's about the heterotoph. On this case, look, what is the whole point of contractile vacuoles in the parameciums? It's always about homeostasis. Keep in mind, contractile vacuole is always about homeostasis. You want to make sure that the internal conditions are limit, that means it's in constant. You know, you want to regulate the water inside. So you don't want to have too much water inside the paramecium, otherwise the paramecium cell will burst, die, and you also don't want to have less. So what is the purpose of the contractile vacuole in the paramecium? It's all about regulating the water inside, okay? So these are the ones for paramecium. Now, since we have talked so much about functions of life, you gotta mention 2. And when it says here state 2, that means most certainly that this will be a 2 points, okay? And here is the most important thing, and please pay attention to this. Let's just say I wrote A, B, C, D, E, F, G. You don't know, so you put a bunch of name there. IB will correct the first 2. But out of these, let's say this one is right, this is wrong, this is right, this is wrong, this is right, this is right, this is right. Here's the bad luck for you. When it comes to IB, we're only allowed to look at the first 2 answers. So even though the third one was right, the second one was wrong, you don't get the 2 points, you only get 1 point. That's why when you're listing names, when it says the word state or the command word that says list, remember, the first words are the ones that count. So if it's 3, it will count the first 3. If it's 4, you count the first 4. They will not allow you to just throw so many bunch of words. That's not how it works. They go according to the number. So in this case, 2 points, only AP will take into consideration, and they will not even look at the rest of the answers. So what are the state functions of life? Nutrition is 1, metabolism, growth, response or irritability, excretion, homeostasis, and reproduction. Now, here's the ugly part. Do not allow feeding. Why? Because plants do not feed. So that's why it's important that you do not use the word feeding as a function of life. You can never say this. You have to use the word nutrition, because plants don't feed on things, right? They do photosynthesis. And many students, I see the word saying, oh, eating food. Well, you can't. Plants don't eat them. I mean, there are some carnivorous plants, but generalizing, plants doing photosynthesis. So do not use the word feed. Use the word nutrition instead. And as I was mentioning, mark the first 2 answers only. So if your first 2 answers are wrong and the rest is right, too bad. So this can be any of these guys. It's your choice. As long as you mention these guys, you get the 2 points. Next question. So we have here unicellular and multicellular organisms share the same functions of life, right? So they're just saying that a one cell and many cells, right, of organisms, they say the same functions of life, which again was nutrition, metabolism, growth, response, homeostasis, all these other things. Agree? Well, here is the thing. The next part, it says outline 4 of the functions. And here's where I always tell students, outline does not mean just list. It does not mean just state, my friend. You have to mention. So you have to say, for example, oh, it grows. What does it mean grow? You can't just say grow. You gotta say, oh, it increases in size. You have extra numbers of cells in there. Or you talk about, let's say, homeostasis. What does homeostasis mean? Oh, they want to make sure the internal environment inside of him is stable. It's maintained, constant. So you have to describe these words. So you are describing 4 functions, right? So there's going to be 4 functions here. But you also have to talk about them, because it's an outline. An outline means go a little bit deeper on information. So let's see. The first one here, what does OWTTE means? It just means other words that take it to the same effect, right? So it doesn't have to be exactly this sentence, process by which organisms take in and make use of foods or nutrients. No, you can say nutrition. Nutrition, you can say it's a process where there's the digestion of the food or is the making of the food by the plants. You can say this. You can also have metabolism, which conversion of organic molecules and chemical reactions in an organism. Now, conversion of what organic molecules? We can talk about, example, glucose is organic, so you are converting it. Or you can talk about chemical reactions, right? Because metabolism is all about all the chemical reactions happening in your body. You can talk about growth, as I have mentioned, increase in size or increase in number of cells. Response, that means how you react to the environment or to a stimuli. Like, for example, if somebody slaps you, well, how you react? Get pissed off or just walk away? That's a response. Next, homeostasis. As I told you, it's about maintaining constant or keep a stable internal environment. Describe what homeostasis is. Reproduction, production of similar cells, organisms from existing ones. Or you can just say reproduce means you are creating new cells. Or you can also say from sperms and eggs, we're making new babies. They will accept that. They're not that picky on this one. And excretion, you just getting eliminations, waste. Some students ask me, Miss, if I wrote pee or pooping, would it take points off? No, not really. We're still going to take into consideration because it still is excretion, because pee and poop are wastes. But some IB examiners I have seen, they're very picky, like, no, I want it scientifically. But don't panic. If you don't remember, just write the common words. You might more likely get the rights to fight for that point. Okay? So these are all the functions of life, but you only have to mention 4 out of all of these. Choose the ones you're most comfortable describing and put it into the test. So here's another one of the questions. It says, outline the reasons for the difference between proteomes of cells within a multicellular organism. So then students are like, what the hell is proteome? Just from the word protein, that's close to the word protein. The here is a study of all the proteins there is in the world. That's what proteome is. It's all the proteins. Now they're saying, what are the reasons for so many different types of proteins in the cells within the body? Now, you got to talk about how you're going from a gene to become so many different types of proteins. So let me here put a little bit more of a drawing so you can, first I can talk about it, make sure that you understand this before we get to the rest of that. So just to be clear, let me take this one out. Usually, let's say you have a DNA here, right? This is DNA, and you have genes 1, 2, 3, 4, 5. And you have this in every cell. This is cell 1, right? You're going to have the same sequence of genes. Doesn't mean the base, but the genes will be the same thing on cell number 2. And you have cell number 3. All the trillion cells have this sequence, right? So what happened is cell 1 might be in your hand. So what it would do, it would probably trigger maybe this guy right here. Now it is on the outside, some kind of communication, and it can be so many, like thousands of different types of stimuli, 1,000 times of different environment, 1,000 times of a trigger that will cause this gene right here to start making a protein. Now here's the thing. You have so many different types of stimuli, which that means just this gene, it can create over 1 million different types of protein, depending on its location and depending on what is triggering him. And then imagine that cell 2, is not located. Let's say this is in the arm and this is in the leg. That same gene is also found on cell 2, but guess what? If this one was in the arm and decided to make protein B, this, because it was in the leg, it decided to make protein A. Different locations, different environments trigger the gene to make different types of proteins. And this is just one gene. Imagine this one, it has another millions of proteins, and this one has a million of proteins and millions of proteins, and it depends on which environment it's exposed to. So there is a huge variety of different types of proteins, and each protein has a different type of function. So that's the general idea about proteins. Now, think about this. How many points? This usually can be 4 points. It's an outline. Outlines will, okay, you will never have an outline that is more than 4 points. If there is, IB has changed its thinking. But usually outlines go maximum to 4 points. After this, it will be anything that is 5 and above. You'll be like describe, discuss, explain, and so forth. So let's look at what the Marxian will say about this. So the first thing in the Marxian, it says that right here, all the genome. And when I say the word genome here, it means all the genes. Just like proteome means all the proteins. So you kind of already get. So here saying all the genome is the same in all cells of the organism. It goes back to what I told you. See cell 1 here has the same genes as cell 2, the same genes, okay? So he's saying that all the genome is the same in all the cells of that organism. Every cell. So if, for example, if you have 100 million cells, all of your 100 million cells, they have exactly the same genome, the same amount of genes. The second thing, here it says the genome or DNA or genes instructs the production of protein or proteome. So like we said in the drawing here, it is because here this gene is what is going to make the protein. So who gives the information? It is the genes. You can say genome, DNA, or gene. What it does, it instructs the production of proteins or the expression of proteins. So genes tell you how to make a protein. Next, the proteome is all proteins produced by the cell. That's what it means. Proteome is all of the proteins inside of a cell. And we said that. Again, proteome is all the proteins inside a cell. Right here, the proteome varies with the function, location, cell differentiation, or the environment conditions. That's what I was talking about right here in the drawing. Depending on where the genes are located. Is it located in the arm? Then you have this. If it's located in the legs, then the gene will produce another type of protein. So that thing that I was just showing you right now is that the proteome, that means the protein will vary depending on where it is located. Where is the gene located? Is the gene located in your head? Is it located in your belly? So that means it will give you a different type of function, right? So if the gene is in a specific environment, that gene will express different than if it was in another one, okay? And that means you express a certain protein in an area. And if you go, so let me just do the drawing here just to make sure if you guys understand. Let's say this gene here is located in area number 1. If this gene is located in area 1, it's going to produce protein B. Now you take this exactly same gene, now you put him in an area that is 2. Guess what? That gene that always produced B, because he switched his area, now he's going to produce A. Now, this is a protein, B is a protein, A is a protein, but these are different types of proteins. So you will produce different types of proteins depending on the location that you are at. Now, we have here specific genes are expressed, turned off in different cells. And it makes sense. Why? What am I saying this? For example, you have these 3 cells, and let's say they all have the same exact genes. And that is true. They have to have the same exact genes, right? But this is trying to say is specific genes expressed in different cells. In this cell, maybe these 3 are sleeping and this one expressed and makes your cell become a heart. Maybe in this one, these 2 are sleeping and these 2 are awake or expressed. This becomes a lung. Maybe on this one, all of these are sleeping and only this one is awake and it makes you a leg. So depending on the gene that is expressing, it will tell you what kind of cell will come out, right? So again, specific genes are expressed in different cells. What I'm trying to say is not all the genes inside a cell are expressed. Some genes will be on, some of them will be off. And last but not least, turning on and off according to the required function. That's obvious. You want to turn this gene because you want that cell to be a heart. So you want that cell to do pumping. Oh, you want to turn that gene because you want that cell to do breathing. So that cell will become a lung. So it's about the function. You just turn on a gene basing on what you want that cell to do. It's what its function is. That's the most important thing is, I'm going to turn on this gene because I want the cell to do some kind of movement. So, recapping, when we talk about proteomes and genomes, first mention that all the genomes are the same in all the cells, right? And the genomes, what are they doing? They are producing protein. And basically, you got to mention what proteome is, which is all the proteins inside the cell. Then you got to say the proteins vary in function depending which environment they're going to be at. And some genes are turned on in certain areas in certain cells, right? And they are turned on in certain areas because they require a specific function. If you have this, my friend, you got all the points in this. So this is when we're talking about proteomes and genomes. It's a very hard question in a sense, like I haven't seen it much, but it could come out. So this is a good one to keep in the basis, especially because if it hasn't been seen a lot, it most likely will fall. Now the next question we have here is outline the significance of surface area to volume ratio in the limitation of cell size. Now this I already have an entire video talking about it, but just to do a quick summary of this, let it be clear that when we talk about surface area, all right, surface area in this situation is the same thing as says the contact with the outside of the cell, okay? And volume has to do with the cytoplasm. That means the space inside the cell. Oops. So again, surface area is how am I in contact with the outside of my cell. Volume is how much space I have inside of the cell. So these two are very important to know the cell size. And we're going to explain this. So imagine you have 3 different cells with 3 different sizes. And cells go through metabolism, so sometimes they produce toxic waste that they need to get rid of. So if you look at this little red dot, it's a toxic. To get out of the first one, it takes him maybe 1 second. This one takes him 5 minutes. And then this takes him 1 hour. Now, the cell can handle a toxic inside of him for that 5 minutes. After 5 minutes, if it's 6 minutes inside, the cell dies. So in this case scenario, the cell is not functioning right, see, because he's not able to get rid of his wastes in a fast manner, right? So remember, the cell is always bringing things in and out of himself. So he always have to be able to do these kind of movements, but he has to do this in a good rate, in a good pace, in a good timing. If you take too long to get rid of something, you're going to get sick. So what happens if you reach that 5 minutes limits? Well, the only thing you can think of, instead of continue growing the cell, what do? You divide. That means you start doing cell division to keep the cells always in a small size. That's what it means by the limitation right here of cell size. Then that's all because of the surface area and volume. What does that mean? Well, okay, I get the point that the toxic has to get out of the cell, but what you're trying to say with the surface area and the volume? What I'm trying to say here is this, is that the volume, think of this, think of volume as trash, and think of surface area as how fast you can get rid of it. If the volume, in this case, keeps getting big, but you are not getting faster as it gets big. See, you continue with a certain pace, right? In getting rid of the trash. But the volume just keeps getting bigger and bigger. That means the trash keeps getting bigger and bigger, but you keep the same pace. What's going to happen? You're going to pile trash inside your house. Does that make sense? So that's not a good sign. So every time volume is increasing, the surface area should be increasing. That's what happens. But sometimes the volume will reach a point that it's so big that the surface area cannot handle taking it all out. And this is the time that you know that the ratio is not good. That means the relationship with these two are not good enough anymore. That means it's time to divide. That's why we say the ratio between them, or the relationship between the surface area and volume, has to be balanced. So when the volume gets too big that the surface area cannot handle taking the stuff out, it's time to make that cell that has become so big become small again. How would you get all of this into mark schemes? Let's look at the mark scheme when it talks about it. First, you gotta mention the surface areas of the cells affects the rate of the material exchange. That's exactly what was mentioned. Remember, the surface area has to do with the rate. Rate here, we're talking about the time. The time to do what? To exchange the material with the outside, or like taking the trash out of the cell, or bringing things inside the cell, right? So the surface area has a big influence on what goes in and out of the cell. Most important, how fast it goes in and out, because that's what the word rate means, the timing it takes it. And the surface areas influence the time it takes things in and out. The next thing we have here is when the cell increases in size, so does the chemical activity. Oh yeah, so imagine if the cell is getting bigger, that means it's doing more functions. And if it's doing more functions, that means there might be more waste. If there is more function, more waste, most likely what? There's going to be a lot more that needs to go out. So you need a better surface area to do this. Now, when the cell increases in size, more substance needs to be taken out, more waste product needs to be excreted. So these two here combine each other, right? As the cell is getting bigger, that means there's more metabolism. So that means there's more waste coming out, which means that means we need to get more of this stuff out. And what did IB do? They split this into 2 points. So just saying, the cell is getting bigger, you having more metabolism, that means you have more things to exchange in and out of the cell. Guess what? We need to work on this. Then it leads to the next one. As the volume of the cell increases, right? Because if the cell is increasing, that means the volume is increasing. So does the surface area increase. However, this is the most important part, not at the same extent. Or as I say, on another way you can say it, because it says here or, when the cell gets bigger, its surface area to volume ratio gets smaller. So the surface area gets smaller. The smaller it is, the worse it's going to get. This is not good. So what are you trying to say is the volume does not increase at the same pace as the surface area. Sometimes the volume will get so big that the surface area cannot handle the pace, and that will be an issue. So with that, it means substance will not be able to enter the cell fast enough. Or you can say the volume will not be supplied, or the metabolic rate will exceed the rate of exchange. So when the surface area and volume ratio is higher, the diffusion rate increases. So that means there's a lot more things going on if the ratio is big, but if the ratio is small, it's not a good sign. So what is this trying to say is if the cell gets bigger, that means the less chance of substance getting in and out, less chances of getting food in, and that means you're going to have problems. So the ratio between the surface area and the volume is very important. And then it says here, some cells have adaptation to increase the surface area, like flatten, microvilli, red blood cells. What are all these saying? This is just an exception. That means some cells, if they're getting bigger, bigger, and bigger, what do they end up doing? They start doing this. They start folding. When they fold, they increase the contact, the surface area. That's what it means. That's what adaptation is. It's just folding the cell membrane so they have more contact with the outside. That means it's increasing the surface area. That's what you just meant. So just mention this part. Some cells have adaptation to increase surface area. This is what it is. It's just making extra folding. So looking at the recap, this is all the information. This will be an outline. This will be like probably a 4 points. But this is not the only thing. There's more. You can say here, cells in growth areas tend to divide and remain small, or cell divide when maximum size is reached. So in conclusion, if the cell is getting too big, it is time for divide. Look at this, how it points. Now, if this was a 4 point, I can say the surface area cells affects the rate in which things are coming in and out. If the cell is getting bigger in size, that means there's more chemical reaction, which that means more substance needs to be taken in or out, And if the volume increases so much, the surface area will increase. However, it's going to reach a point that is not going to be good enough, right? And if this happens, the cell right here tends to divide and remain small. Boom, that's 4 points. You don't even have to mention all of this, but you have to look at the 4 points. So these guys, it's extremely important that you know how to describe all of this, because it might be more, it might be 5, it might be 6. But you see, there's a large point here. Remember, the volume has to do with the amount of metabolism there's inside. The surface areas has to do with the exchange of inside and outside, and they should be in a good ratio. However, if the volume keeps increasing, it's going to reach a point where the surface area is not going to be able to take up with that, and then the cell will have to divide. Otherwise, the cell will die. So this is a good one, and this is something that is very, very common question. Why am I saying common? You will find this question for sure happening as a multiple choice, and if it's not there, it's probably going to show up as a paper 2 question. Now, this is just an outline. Here is another question about surface area to volume that influence. This question, look very careful. It's exactly the same thing as this one. Look, this says outline the significance of surface to volume ratio and the limitation of cell size. And this one says explain how the surface area to ratio influence the cell size. It is the same question. However, there's one thing that is different, and is this right here, explain. And this one is outline. Now look at the mark scheme. We're going to look at the mark scheme, and I'm going to show you how many things are in common. But just because it's explained, that means that you have to explain more in details. Our outline is very generalized. So the first one, it says here, small cells have larger ratio than larger cells, or the ratio decreases as the size increases. That was there, right? It was here. It was talking about here, the ratio getting smaller, right? And then here you can say that the ratio will get if it's higher. So the point is that's a big one. So if you see it common in 2 mark scheme, make sure you talk about the ratio. So larger ratio, that means that the size has been increased. Okay? Again, just to make sure it's written here, higher ratio, it means the size is big. Low ratio, it means the size is small. So what do you want to do? Always want to make sure you have a small one, because that means you have more exchanges to happen. The second part, it says surface area membrane must be large enough to absorb nutrients. Again, we did talk. It says you need to have a surface area that is large. That means why? Because remember, surface area is all about exchanging. So the larger the surface area, the more exchange it is. So you're fine bringing in nutrients, and you're fine taking away waste. Next, surface area membrane must be large enough to excrete. Look, in that previous one, they mentioned both together as 1 point. Here, they're putting as 2 points. So you want the surface area to be good in order to get nutrients in, but you also want the surface area to be large in order to get rid of, excrete things out. That's 2 points in this one. Now, need for materials is determined by the cell volume. That means if the volume or the cytoplasm of the cell is getting bigger, that means there's more metabolism. If you have more metabolism, that means you need more nutrients to do the work. So as this volume is increasing, you need more nutrients. As the volume is increasing, there's more metabolism. Cell size is limited. That means cells divide when they reach a certain size. This is so simple, right? Just saying, the cell got too big, time to divide, otherwise it can't function. Then you can talk about diffusion across the membrane of the surface area. What does that mean? When they say reference, they're basically saying, well, if the volume is getting too big, while the surface area is not keeping up, you're not going to have enough time to get nutrients in or to get the waste out. So that means it will disrupt the process, the metabolism of a volume or a metabolism of the cytoplasm of the cell. So that will cause problems. So that's pretty much. Now, on this mark scheme, you can see that it's very brief, right? Even though it's explained. And this one is a lot more in detail. If you go back here, this one has a lot more too. So what do? Focus on this one. Build it up. This, make sure you write. But if you notice, a lot of the wordings they have together is exactly the same. You're just talking what surface area is about, talking about the volume, talk about the exchange of wastes and nutrients in and out, talk about the size, having to make sure that it's in the same size, a low size, because if it gets too big, it has to divide. So that's what the surface area or volume ratio. And don't forget to mention about the ratio, right? If it's too big, gotta divide. If it's too small, it's okay. So we're going to the next one. We're talking now about stem cells. How does the stem cells work? So here we have two questions, and these two questions are questions related to Stargardt disease, which is one of the diseases you must know when it comes to stem cells. So you haven't learned this? Go back to the lecture video. But by now you kind of have an idea. You know that Stargardt disease is a genetic disease. That means it's passing on from parents to the child. We know it's a recessive disease. We also know this disease, it causes people to become blind, and that's because they have those retinal cells in their eyes that gets destroyed. So they lack production of vitamin A and they just destroyed. The good news is that we have stem cells, and we can turn these stem cells into retina cells and replace the old destroyed retina cells, and then the person can go back to have visions. So that's a very quickly oral explanation. But let's see how the mark schemes up. But first, let's read the two questions and see how these two questions are very similar. The first one says, outline the use of human embryonic stem cells to treat Stargardt disease. So here it's focused on what? It's focusing on stem cells, embryonic stem cells. How do they treat it? Here it says stem cells can be used to treat Stargardt disease. State one other condition treated using stem cells. So those questions are talking about Stargardt disease, but one is telling you to explain how stem cells are used in the disease. The other one is saying, well, we use stem cells in Stargardt disease. Where else could we do? So they are talking about the same thing, but they're not asking the same thing. So you got to be very careful with these questions. So let's look at the first thing. Here it says that this is inherent, as I told you. Now, you don't have to use the word degeneration. It's a big word. You can just say it's the breaking down of the retina cells. You can say it's eye genetic. You have to mention that it's genetic, that is inherited, okay? This is a disease that's passed on, okay? Down here, you already have the answer. Leukemia is another treatment of a disease that you can do, or skin burns. You can replace skin by using stem cells. But let's continue here. You also can say the stem cells can provide or differentiate into healthy retina cells. So what I was saying orally, the stem cells can be stimulated to become retina cells, and then these retina cells replace the old retina cells. And then the person go back to being what? With full vision. Another thing is injecting stem cells into the retina or the eye to restore vision. That's exactly what I was just mentioned there. So the first thing you say, it is a genetic disease. Mention what the Stargardt disease is about. It's a genetic disease. What does it do? It destroys the retina cells. Then you talk about stem cells. What is the point? Stem cells can be differentiated into those retina cells, and then they can put it in the eye to restore the vision. That's 3 points right there. So this is usually an outline, and since outline maximum is 4 points, you can easily mention these three easily, okay? So with that, we're done for this one. Let's talk about the use of adult stem cells. We are talking here about what are the advantage and disadvantage of stem cells. Now, this is an essay that can be very much seen on paper 2 on the section B. This is the discuss. And since the word discuss, you will never see something discussed with just points like 4 points. Nah, not even a 5. When we say discuss, we're talking about 6 to 8 points because you got to talk a lot. It's like writing an essay. Discuss means more like a debate. Talk about the goods and the bad. And they already mentioned here, talking about the good and the bad things when you're using stem cells. Now, there's a huge list out there of why stem cells are good and why stem cells are bad. Now, if you still don't remember about stem cells, please go back to that video on stem cells and learn about it. Learn how to use them, what is their traits, and what kind of diseases they can cure or treat. So let's look at the mark scheme. So the first thing is that you can mention when it comes to stem cells is tell what the stem cells is all about. Stem cells can divide endlessly. That means they can differentiate. Now, here is where students don't get it. It mentioned adult stem cells. It did not mention embryonic stem cells, which is a completely different story, okay? Adult stem cells, They already have been positions for, like, such as in the bone marrow, it can be on your nose, but they're not what we say as a pluripotent or totipotent, which these guys basically can be anything. So that's the first one. Second, they can be used to repair or generate tissues. So just these two, the first two, we're mentioning what stem cells is. Stem cells can differentiate and stem cells can repair tissues. That's 2 points. And I see these two answers always when it comes to what stem cells are. So make sure you know those two for sure. Mention what stem cells and the other one is. Next, refute ethical objections. Why? Because when you take a stem cell from an adult, that's okay. You're not killing the adult. But if you are taking a stem cell from a baby, that means before it's born, when it's still an embryo, you are killing that baby. That's why it's okay to take stem cells from the bone of an adult. It's okay to take a stem cell out of his nose. Adults give informed consent to use. Now, this is common sense. You don't need this on a book, but it's obviously, if you're going to take a stem cell of an adult, you just say, hey, dude, I need your stem cell. He's going to say no or yes. But you can't go all the way to the embryo and say, hey, little cells, can you just let me take you? It's not going to happen, dude. So this is an advantage that you can do with the adult. The next one here is adult's source is not killed. I already mentioned that. With adult, you don't kill it, but with the embryos, you're definitely going to be killing it. There's no rejection problems. What does that mean? See, let's say you adult, you want to take a stem cell from his bone marrow, but you're taking that stem cell from his bone. So that means it is his stem cell. That means it's his DNA. That means it's his cell. So the body, believe it or not, the body is very picky about this. The body is like, I only accept what I know. If I am accepting something that is a stranger, I will kill it. So if you're taking a stem cell from the adult to make something that is back to him, to his body, he probably will accept it. There's not going to be any rejection problems. So for example, if I take a stem cell from my nose to create a part of my nose, my body is not going to reject that stem cell because it's still mine. But if I take the stem cell from a stranger's nose and put it on me, my body will reject because it's going to say, who the hell are you? So body is very picky with his friends. Next, less chance of cancer. See, there is more chances of playing with the stem cells from the embryo because you're manipulating things. And at the same time, there might be rejections and the body might react in a bad way. But when you're doing it with adults, the chances of cancers is so little. And last, most tissues in adults contain some stem cells. Not most tissues, but there are. So you can still find some stem cells in adult. So these are all the advantage for the adult stem cells. We're not talking about embryonic. Now, it says the advantage and disadvantage. It is very, very important. Let's look at this. If it says 6 points, there always has to be a balance. Do not put 5 advantage and 1 disadvantage. That's not a good sign. Make sure that it's always balanced. It can be a 4-2 or it can be a 3-3. Do not do this because sometimes IB will be picky and say, hey, there has to be at least 2 of each side. So always have at least 2 of one of those sides, the good or the bad, okay? So let's look at the disadvantage. Disadvantage is it's difficult to obtain, collect, or find adult body, very few available. So even though here it says it can be found in most adults' tissues, it's not that easy to find them. They can be there, but doesn't mean you can find them. Second, some adult tissues contain no stem cells. So just because we say there is some adult stem cells, it doesn't mean that you can easily have them. Some adults don't have any more stem cells while others do. So it depends on the body. And last, adult stem cells differentiate into fewer cell types than the embryonic. What does that mean? This is basically saying that, for example, if I take a stem cell from my bone marrow, right? That stem cell can only be doing anything related to the bone. I cannot take this stem cell here and just start to make a heart Why? Because this stem cell has been a little bit differentiated. It's more specific now. Oh, I am a stem cell, but I can become any kind of bone cell. I just cannot become something else. But if that stem cell is taken from an embryo, it's a totally different story. If a stem cell is taken from an embryo, it can be a heart, it can be a lung, it can be a liver, it can be a bone. That means there is more options. That's the difference. So here, this is a big, huge assay we're talking about here. And you have to be very careful in how you're writing it. So recap. First thing is mention what is stem cell. Stem cells can divide and differentiate, and they can repair. Then you talk about ethical. There is no ethical issues with humans, adults. You can ask the permission for the adult to give it. The adult will not be killed. There is no rejections if you take a stem cell and place it within yourself. Less chances of cancer. And you can find stem cells in most of the issues. But the bad news, it's difficult to get it because there's very few of them available, and some adults don't even have it. And the adult stem cells is already differentiated, so you cannot turn into any kind of cell like the embryonic cells. And that's pretty much what we get with the assays for stem cells. But we have many other questions like these. This one says, outline differentiation of cells in multicellular organisms, which is a 4 marks. And you have this one that says, describe the importance of stem cells in differentiation. Now, what do we see here? These two questions are basically asking the same thing, but it's worded differently. First, the command is different. This is outline, this is describe. But ironically, this has 4 marks and this is at 3 marks. Okay? This one is basically saying, right here, oops, let me go to the highlight, is saying, outline the differentiation of cells in multicells. So we're asking, how are the cells differentiated? And here we're saying, how important stem cells in differentiation. So indirectly, they're talking about how stem cells are differentiated. Let's look at their mark scheme. We have it here. The first one saying differentiation is development of development in different specific ways. That means the cells are coming to become different functions. Now, here it says stem cells are undifferentiated cells. It's true. Stem cells have not been differentiated, but they can differentiate into anything. Next, cells carry out specialized functions. They become specialized. That's when they are differentiated. Embryo cells are stem cells. So far here, we're just describing what stem cells are. And this one, we're actually explaining about the differentiation. Third, example of differentiated cell in a multicellular organism. Example, heart cell, lung cell, bone cell. Here, stem cells can differentiate in many ways. They are pluripotent. So if you notice what the difference was, this described the importance of stem cells. That means, what is it about stem cells that makes it so important for her? So it's because they're undifferentiated. It's because these guys basically are stem cells, which are their embryo, and because they can become anything and they're pluripotent. This case is outline differentiation of cells. The differentiation means they become a specific job, they have a certain function, and again, give an example like the heart. Continue. We have here, cells have all genes, could develop in any way. So that means we know that all the cells have genes, but depending where that gene is exposed to which environment it is, that's the expression it will have. That's what the gene is going to become into. So it can be written this way, but in another way, and I still get points. On this side, differentiation involves expressing some genes but not others. These guys are very close related. That means we have all the genes in every cell. We have exactly the same genes, but the way they're differentiated, it really depends on the location of that cell. And here says some genes are switched, expressed, but others are not. Again, just repeating. Some genes will be expressed because in that location, but if they move to another place, they will be shut down and other genes will be expressing. And on this side, stem cells can be used to repair, replace tissues. So again, this is just describing what the stem cells. So if you see a common trend here is, first start explaining what stem cells are, right? They can be repaired, stem cells are undifferentiated, they are used to treat whatever diseases. Now, where are they located? They're located in the embryo. You can find sometimes in adult stem cells. And then after you mention this, then talk about differentiation. What is differentiation? Well, differentiation means some cells are triggers while others are not, depending on the location. And by being triggered, you get a certain function. And once you differentiate it, you can't much do other things besides that function. So leading to the next one here, we have the position, the hormone, cell-to-cell signal chemical determine how the cell develop. This is again just saying, where are you located at? If you're located at the arm, you're going to do a certain type of gene. If you're located at the legs, you're going to do a certain type of gene. And then we can continue adding more, but such as a group of differentiated cells is a tissue. That makes sense, right? If it's a heart tissue, that means these cells of the heart have been differentiated to become a heart cells. If it's a bunch of cells in a lung, it's a lung tissue, and this lung tissue has a bunch of differentiated cells that will basically they have become a lung function. So these are just common sense, but again, when you're going to write an essay about stem cells, what's the first thing? Mention the two traits, right? The first thing is that they're undifferentiated, and mention that they can be differentiated and they can replicate themselves, right? Those are the two. Then talk about what differentiation means. That means depends on the environment, and the environment has a certain genes that are expressed, and if there's those genes expressed, we have a certain function. And then you talk about location. Where do you find the stem cells? You can find it in the embryo, or you can find in adult stem cells in other parts like the bone marrow. And then what is it used for? You can also use them to repair or to treat diseases. So these are the things that you have to mention when it comes to stem cell. If you mention these, you're good to go. So now we have this question. This question here is talking about electron microscope and light microscope, which is still part of 1.1. So the first question is reading out. So it says here, the figure shows the transmission of electron micrograph of a retrovirus particle. Now it's 70 nanometers in diameter. Now state a reason for using electron microscope to view this virus rather than a light microscope. When you want to answer this question, you have to remember the difference between light and electron microscope. Now, we know that the light one is not expensive while the electron is, but this one you can see it 5 times more, when this one you can go for 200 times bigger. So this one, it has a bigger resolution. That means you can see it more clearly than this one. This you can see living ones as well as dead cells, right? Well, this one you can only see dead cells. Now here it says that this guy is 70 nanometers. That is very, very tiny. So if I would be looking to see something that's extremely tiny as nanometers, it's not even micrometers, just forget it. Micrometers is bigger than nanometers. I would definitely be using the electron ones because I just want to see it, right? So why would I be using the electron one? Just because its resolution is bigger. That means you can see the picture so much clear. And another way, viruses are too tiny and too tiny to be seen in a light microscope. So you can see virus in electron. Obviously, very rare that you will be able to see a virus in a microscope. If we could do this, woohoo, it would have been easy to see in high school, but that's not true. So viruses are very small, and since they're very small, they can only be seen through the electron. And we have it here, see? They're greater resolution, and that means they're too small to be viewed by the light microscope. Again, nanometers, too tiny for light microscope. When you see nanometers, automatically go to the electron. And that's pretty much of the questions that are most common seen for 1.1. But here are some of the questions that haven't seen yet in IB exams. They might pop out, but I'm just putting here just for the sake of it. Maybe go for it, search for it. So just take a pause in the video and read these questions, because some of these questions, you never know, might just show up in the future. And these are stuff that actually is in the content, but I haven't seen them put much of these questions in IB. So these are just me taking future guesses of questions that might show up. You take a pause and you can look at this. You never know, it might just show up. And that's all for the written answers on topic 1.1.